Well, good afternoon, or good morning, or good evening, wherever you are. It is my great pleasure to welcome you to this seminar in the series of the Department of Epidemiology of the Harvard School of Public Health. And it is my distinct pleasure to introduce our speaker of today, Dr. Jordan Smoller. Dr. Smoller is a professor of psychiatry at Harvard Medical School and a professor of epidemiology with us also at the Harvard School of Public Health. He is the Gerald Rosenbaum Endowed Chair in Psychiatry Professor at MGH, the Director of the Center for Precision Psychiatry, and the Director of the Psychiatric and Neurodevelopmental Genetics Unit in the Center for Genomic Medicine. This is quite something. He is a psychiatrist, epidemiologist, and geneticist. A brilliant combination. You cannot have it better, Jordan. Thank you very much for doing this. And he studies genetic and environmental determinants of psychiatric disorders across the lifespan. And he will show examples, I'm sure of that. It's a great pleasure to have you here, Jordan. And it's my privilege to give you the floor to talk about cross-disorder genetics of psychiatric disorders. Please go ahead.
Thank you so much, Bert. It's great to be here, and I really appreciate the invitation. And when you reached out about potentially speaking about some of the work we've done recently in cross-disorder genetics of psychiatry, I was thrilled to say yes, not least because it is, of course, the Department of Epidemiology, in which I'm not only, as you mentioned, a faculty member, but did my training in epidemiology, and I see some of the folks here were instrumental in helping me become an epidemiologist. So
Yeah,
that
thank you.
that is a wonderful treat as well. So I'm going to bring up this talk and start us off. So let's see if I can do that here. Okay. Are you seeing it?
We see it, but not yet in the present. Yeah,
There you go.
now we
How
see
about
it.
that?
Well,
All right.
wonderful. Thanks. Yeah.
Okay, so where I'm going to start is just right in the middle of the fact that I'll take as given that we understand that psychiatric disorders are very common conditions. They are associated with tremendous morbidity, but also mortality, early mortality, both from chronic medical illnesses that are associated with them, from suicide, and so on, and that our understanding of what causes them has been progressing, although somewhat challenged. And one area that there's obviously been a lot of interest in is in understanding the potential genetic basis. And here, I would say we have had a lot of progress in just the last, 15 or so years. So what you're looking at here is a timeline across the bottom of years, and then on the y-axis, the number of risk variants or significant associations that have been identified with DNA variations with psychiatric disorders. That little inset bar graph is just a reminder that these are conditions for which the heritability, that is, the impact of individual differences in our genetics on what, how these conditions are distributed in the population is substantial for all of the psychiatric disorders. And then on the right, that little inset table is just sort of showing you the number of loci that have already been reported for a whole range of these conditions. So this is, I think, really kind of a success. You'll see that inflection point happening around 2009, 2010. That's really when not only had the technology advanced to allow us to do genome-wide studies, and we had a map of the human genome, but there was tremendous scientific collaboration that really began around then. The Psychiatric Genomics Consortium in our field was it was really been the leader, pulling together researchers from around the world to really build studies that enable us to fairly robustly identify risk loci. So that's been great. And for example, for schizophrenia, this is just a recent snapshot of the sort of genetic architecture or polygenic components that have been identified. And again, on the y-axis, you see the odds ratio associated with the variation in whatever it is you're looking at there in the plot. And then on the bottom, the allele frequency in the population, how common is that variation in the population? And the different colors represent different kinds of variation. So this is true, we think, for most psychiatric disorders, that they are highly polygenic. That is, that multiple, maybe hundreds or thousands of genetic variations could be involved in contributing in any given situation, and that they range across the allele frequency spectrum, meaning they could be very common, with most people carrying uh, some of these, or rare and even de novo, not inherited, but arising in the gametes or the embryo. So the really strongest one you see up there, the 22q11 deletion, is a copy number variation that's highly associated with risk of schizophrenia, psychotic illness. And there are now a number of single gene variations. Actually, these are all copy number variations, these sort of in yellow or orange, that have been identified. Um, this smudge actually represents common variation that is just barely peaking above the null line there. But this is where a lot of the variation is, in these small variants that each contribute individually a small amount. So this is sort of the complicated picture that you see evolving even just for one disorder. And more than just sort of identifying a list of these variations, the payoff is sort of when they begin to tell us biological stories or insights that we might not even have thought about before. So these are some of the themes that have emerged when we look at what are the genes or the variants that are involved actually related to. Are there pathways? Are there sets of genes that seem to be working together? And that, in fact, is the case. We're beginning to see some of these, and I'm not going to go into detail on these, but you get the idea. Okay, so that's great. We've seen tremendous progress. Now, for those who don't necessarily think about psychiatry all the time, um, we haven't talked really about the phenotype. And in psychiatry, the phenotype is the trickiest part in many ways. And the way we decide whether somebody has a disorder in clinical practice and typically in research is to determine whether they meet the diagnostic criteria for that condition. And those diagnostic criteria, the reference we typically use is the Diagnostic and Statistical Manual, the DSM. And this is a book which lays out diagnostic criteria. It's a book that has been in publication with various editions since 1952. And what you're seeing here is the rising number of diagnostic labels in that book, which reflects typically a greater splitting up of diagnoses into more fine-grained sub-diagnoses or subtypes. But even if you just look at sort of a subset of these, you can see the amount of reshuffling, splitting, and lumping that goes on. And I'm just going to show you that in a little timeline here. And what you're going to see across the bottom are the different editions, starting with DSM-3, which is really where this all sort of started, our modern system, and going up to DSM-5. And we're going to look at sort of pervasive developmental disorder type conditions, mood disorders, anxiety disorders. And so here's what you see. With DSM-3, you've got these three categories and you've got a bunch of disorders in them. And then as revisions of this book go on, you see disorders that, drop in. Some are disappearing, some are changing their names. And then when you get to DSM-5, an unusual thing happens, which is instead of just splitting, you see some lumping there on the autism spectrum disorder side. And then some of the things that were in the anxiety disorder category actually move into whole new categories that we didn't even have before, uh, particularly trauma and stress related disorders and sort of more uh, obsessive compulsive disorders. So the point being, this is a moving target. And the way we make these diagnostic distinctions is essentially by a consensus of experts evaluating the latest, best description. It's without respect to etiology. And that was by design, meaning people are not placing bets yet on what causes these things. And so we don't have as part of it even the pathogenesis and that was deliberate in DSM-3 because at the time, and this is actually still true, people thought what we really want to do is emphasize reliability of these diagnoses because the reliability up to that point had been really poor. That's going to be a prerequisite for their validity, but we don't want to say that we know what's causing them because we don't really know. And if we start getting into those fights about whether it's, early childhood psychosexual development or some brain region, we're just going to be arguing and not get anywhere. So that was totally understandable. Now, um, what happened though is everybody recognizes that these diagnostic categories are sort of provisional. They are heuristic, if you will, although they have tremendous clinical impact. But for example, comorbidity is the rule. So most people with a psychiatric disorder will be diagnosable with another psychiatric disorder by far. And the drug treatments, for example, that we have for treating a range of these conditions are fairly non-specific. So the SSRI, SNRI antidepressants, which people are probably familiar with, like, you know, uh, Prozac kinds of medicines, they've been used effectively for, in some cases, bipolar disorder, depression, anxiety, OCD, eating disorders, post-traumatic stress disorder. So there's a lot of overlap. And that raises the question of what's there. I like to just show this because one of the people, my mentor, actually, my advisor at HSPH, when I was getting my master's and doctorate, in addition to Deborah Blacker, who's on here on the call, I know, Ming Swan, who, along with Steve Ferrone, Mike Lyons, was very interested in this question of phenotype and psychiatric genetics. And this is from a paper of theirs referencing Ken Kendler, who introduced the term scientific nosology to describe nosologic symptoms, nosology meaning systems of classification that are created by formulating and testing hypotheses. We use the term psychiatric genetic nosology to refer to a scientific nosology created from psychiatric genetic data that seeks to classify patients into categories that correspond to distinct genetic entities. That was actually an influential sort of concept for me, although, uh, you know, I, I think, and they go on to say this isn't meant to substitute for clinical practice or anything like that, but it turns out to be complicated as they probably anticipated. So one of the first big efforts was through the Psychiatric Genomics Consortium to look at a DNA level, how much overlap could we see? And this was our first big effort to do this. At the time, we had 33,000 cases across 5 conditions: autism, ADHD, depression, bipolar disorder, schizophrenia. And in the first instance, we looked at could we identify any variants or any genes and variants that might be associated with more than one of these conditions, might be what we would call pleiotropic. And we did, in fact, identify such variants. You're seeing that on the left. And then what about at the aggregate level? Could we look at a genome-wide level, essentially? How correlated are these conditions? And this was one of the first studies that really used that kind of approach, and it used a method called GCTA, which is still used. Uh, what we found was the correlation in some cases among these conditions, which we think of as clinically distinct, or at least the DSM does, were sometimes quite high. So look at bipolar disorder and schizophrenia, 0.68. That's notable because those conditions are treated as mutually exclusive in the DSM. You can't have both. And in some ways, they are the canonical diagnostic distinction that our modern system of classification derives from. There was a famous psychiatrist named Kreplin, 100 and whatever, 20 or 30 years ago, who really identified these two as distinctive conditions. And yet, at a biological level or a genetic level, at least, there seemed to be quite a bit of overlap. one of the advances that has really accelerated this area is the development of statistical methods that allow us to estimate a number of things relevant to this idea of cross-disorder genetic relationships. And I'm not going to go into great detail here. One of them developed about 10, 12 years ago, including Alvis Price, you see there, and uh, others. Uh, is something called LD score regression. And again, I'm not going to go into this in great detail, but what it allows you to do is to take summary statistics from genome-wide association studies. So you don't have to have the individual level data and compute a number of very useful um, uh, estimates. And this has been extended in a number of other directions. But one of them is you can compute the SNP heritability, that is the proportion of phenotypic variance that's attributable to variation in these common single nucleotide polymorphisms. But you can also estimate the genetic correlation, again, using summary statistics and pulling data together from multiple genome-wide association studies. And that is both computationally more efficient than what was uh, possible before, and because of the use of summary statistics, has become very widely used. So that then became available to us in a number of studies. And this is a study by the Brainstorm Consortium, which involved a lot of people here in Boston as well as around the world. And it sort of looked at what is the genetic relationship, genetic correlation among disorders that we think of as psychiatric and disorders we think of as neurologic, and to what extent do they overlap? And this was a very large study at the time, still a large study. Uh, and what you can see there are heat maps, essentially, of those relationships. And the darker the blue, the stronger the genetic correlation, and those asterisks represent statistically significant estimates. And I think the takeaway here was there was more blue in psychiatry than in perhaps neurology. But when you put them together, there wasn't that much genetic overlap, which was kind of surprising in a way, because we often think of, this artificial distinction, let's say, between psychiatry and neurology at a biological level. It's all the brain. But, this provided some evidence that at least at that time, we didn't see a ton of genetic correlation across these domains. And then another statistical method that was important for this future work that I'll talk about was the development of something called genomic structural equation modeling, led by a number of people, Andrew Grotzinger, who used to be here and at this time was in Texas, with Elliot Tucker-Drobe. And this is a very flexible framework, as you can see here, for modeling um, genetic associations across multiple traits, let's say. So a multivariate relationship at the genetic level. And it uses LD score regression to create a genetic covariance matrix that then can fit a user-specified structural model of the relationships of these elements, and then sort of evaluate how good is the fit of that model. We'll talk a bit more about this. So with those advances in hand, this was our second cross-disorder group analysis. Now in 2019, we added 200,000 more cases and three more conditions, and expanded the sample sizes. And here you're seeing the genetic correlations represented as kind of a network map. Again, 0.70, you probably can't see that, between bipolar disorder and schizophrenia, so pretty stable there. We've now added anorexia nervosa, OCD, obsessive compulsive disorder, and Tourette syndrome. And when we did a genome-wide association study, found over 100 loci that were associated with at least two of these conditions. And then we apply this genomic structural equation modeling. And what this does is, so you can see here these 8 conditions that I mentioned. And this is a structural equation model. And these are latent genomic factors that are essentially accounting for or on which these different individual disorders are kind of loading, um, and that are capturing most of the variance. And so you see here that a model in which we have these 3 latent factors, fit was the sort of the best fitting model. And we named them, the compulsive disorders factor, the mood psychotic disorders factor, and the early neurodevelopmental factor, largely because of just sort of heuristics. So anorexia, OCD, and Tourette syndrome, despite the fact that they're in different chapters in the book of the DSM, all sort of are loading and sort of captured by this latent factor. They all have in common a kind of compulsive disorder component. And then we have schizophrenia, bipolar disorder, and some of depression, although not as much. And then, ADHD, autism, and Tourette. We also were able to look at the expression of these pleiotropic loci in the brain, in postmortem brain, and found that though the pleiotropic loci compared to other loci seem to have heightened expression in sort of the mid uh, gestational period, and they stayed fairly high compared to other loci. So there were some very interesting clues. There's a lot more in that paper I won't go into. But there were some loci that were associated with multiple disorders. This one, DCC, which is actually stands for deleted and colorectal cancer, was associated with all 8 of the disorders we looked at. And you can imagine different mechanisms by which a gene, let's say, might be involved in multiple clinically recognized syndromes. They can include things like isoform diversity. Here's the neurexin-1 gene. Or they might be a transcription factor that regulates a lot of downstream effects. DCC that I mentioned is a master regulator of axonal guidance, so the development of white matter in the brain. RB-FOX1, in other words, was very highly pleiotropic, regulates tissue-specific alternative splicing that's involved in neuronal development. So we start to get a picture of biology. A couple of subsequent studies have looked at this genomic structure in slightly different ways. So here's one looking at introducing a method called genomic structural invariance testing and asking essentially, is the structure uh, different between the sexes, between male and female? And the answer here was this isn't exactly the same set of disorders, but it's highly overlapping. But the point was there was not much uh, difference in the structure. And that's consistent with prior studies that have looked at genome-wide sex differences. There are some sex differences, but they're not very pervasive so far. And then this one, we were able to add a fourth latent factor, or a fourth latent factor emerged with the addition of some additional disorders. We're now up to 11 disorders. So alcohol, PTSD, and anxiety. Okay. And now get to the latest work. And this is, as you can see, we started with 33,000 cases. We're now up to 1.6 million cases. And now what we wanted to do, we had 14 different disorders. And there had been all this methods development, and we thought maybe we could triangulate these methods, or I guess it's a higher dimension than triangulate, but we could apply multiple methods that are sort of getting at different things and providing different insights to see, can we get a more complete picture of the relationships, both disorder specific and shared among these conditions. And so again, we now have 14 conditions. We've added a variety of other things: opioid use disorder, cannabis, nicotine, um, what else was new here? Uh, I think, and alcohol is here as well. And so we looked at multiple levels of analysis. Genome-wide, I've already talked about genomic structural equation modeling. Another method called cross-trait mixer, which actually is sort of similar in intent in a way, but it's looking at the degree of overall polygenic overlap and the likely proportion of shared causal variants. This is a cross-ancestry analysis of genetic correlation. There's also methods now to look at the regional correlation, not at the overall genome, but at chromosomal regions. The shared effects are likely to be unevenly distributed across the genome. And then, of course, we can look, if we have these factors, a genome-wide association study. and then apply a method called CCGWAS. This is developed by Voter, Peyrote, and Alcus Price, that essentially looks at the differences in allele frequencies between cases from pairs of disorder. How different are they? And then a whole suite of functional genomic follow-up to sort of get at what's going on biologically and in terms of cell types with the and just highlighting some of the many people who contribute to this effort, particularly the analysts in this first row who really did a tremendous amount, and as well as the folks here and Ken Kendler, with whom I co-chaired the group. Um, Not going to go through this, but we've got a lot of samples, as I said already. And this was the result at the genomic level of this genomic structural equation modeling. So arguably the best fit model was this correlated factors model. Remember, we had these first three essentially from the beginning. We now have these two others. And again, we've labeled them according to their predominant sort of phenotypic features. Schizophrenia and bipolar disorder is now kind of resolved into its own factor. And when you're looking at this, so again, you have these factors, latent factors. These paths have, essentially the relationships, the strength of the correlations between these factors. And then you can see for each of the disorders here, essentially the proportion of variance accounted for by each of the factors. And then this is sort of residual in purple here, residual unshared variance. So that was a very well-fit model, very consistent with what we had done. We also tested another model which fit also adequately, which incorporated what we call the P factor. And the P factor, not invented by us, but this is a concept of a shared fully transdiagnostic vulnerability at a genetic level. So this is a second order factor above these correlated factors. Interestingly, though, if you look at this, the more than 90% of the variance in P is shared with the internalizing factor. So it seems to be largely capturing that. For four well-powered disorders, I'll just show you a little bit of the LAVA analysis of the regional genetic correlation. Basically, 99% of the regional genetic correlations were positive, the same direction, basically, between two disorders. And this mixer method that I talked to you about, this incorporates not only the same directional effects, but potentially opposite directional genetic effects. Most of it by far, though, was in the same direction. That is, let's say variants that influence bipolar disorder and depression were influencing them in the same direction. When we applied that local genetic regional variation kind of analysis, there were a number of specific regions in the genome that seemed to be highly pleiotropic or harbor a lot of this shared variance. You can see the kind of network of them here. One of them on chromosome 11 was what we call a real hotspot because it had significant genetic correlations for 8 different disorders, most of the disorders we looked at. And if you actually look at that region, there's a part of it here that has been very widely studied and implicated in a number of psychiatric phenotypes. It includes the dopamine D2 receptor, which is a target of antipsychotics. It's also been implicated in substance use in the past. So this seems to have a lot of action here, basically. And when we did the multivariate GWAS, essentially doing a GWAS on the factors, we found, 238 unique pleiotropic loci associated with the factors. And then we were trying to look at what about the things that don't fit the factors that might be more disorder specific. And we approach this in two ways. One using a test that comes out of that genomic structural equation modeling, which is called the QSNP test, essentially identifying SNPs that don't load on those genetic factors, don't seem to operate through the factors, and CCGWAS I mentioned before. And the bottom line was the P factor had a lot of uh, Q SNPs, ones that didn't really load well when that was the structure, but the correlated factors model had many fewer. Most of the loci that were identified as being different by either CCGWAS or QSNPs were found between factors. That is, they were loci that influenced disorders on different factors, not within factors. So it sort of, again, reinforces that factor structure. Then we went on to do a whole bunch of functional genomic analyses. And we can talk more about this if there's time, but I realize we have time limits here. But this is, again, looking at different ways of identifying for these genes that emerge from these analyses, where are they active? When are they active? What kinds of biological systems are they implicating? And so here's an example looking at P factor genes versus genes in that were part of this schizophrenia bipolar factor. And one of the interesting findings here is that these putative P factor genes seem to be involved in very general processes, things very pretty fundamental, regulation of RNA metabolic processes, DNA transcription, et cetera. Whereas when you get down to the level of the factor, more specific neuronal brain development kinds of things. Also, um, again, somewhat similar to what we had seen in that prior paper, these sort of factor level or pleiotropic genes seem to be highly expressed, more so than the sort of disorder-specific variants or genes implicated in those pretty early in life and fetal development. That the schizophrenia bipolar factor genes were enriched within excitatory neuronal subtypes in both fetal brain and adult brain. Whereas for the internalizing factor, which again was depression, anxiety, post-traumatic stress disorder, some enrichment in excitatory maturing neurons in fetal brain, but oligodendrocytes fairly uniquely in adult brain. The P factor didn't really pan out as pointing to any specific cell type. And then using another extension of genomic structural equation modeling called stratified genomic structural equation modeling, which essentially looks at what component of the functional annotation accounts for a lot of the signal. Once again, the schizophrenia bipolar factor specific ones were implicating excitatory prefrontal and hippocampal neurons, where again, the internalizing more oligodendrocyte precursors. And again, and then move on to that. So just to summarize what I said so far here, we got these 5 latent factors that captured about 66% of the variance across these 14 disorders. There are some, it's not, the sharing is not uniformly distributed across the chromosomes, but there are some hotspots that seem to harbor a lot of these pleiotropic loci. And we can identify significant loci at the factor level, and they point us to somewhat specific biology, although admittedly not that specific yet. But interestingly, they are starting to point in this direction of a model in which there's maybe there is a sort of more, at least partly broadly transdiagnostic component, depending on how much stock you put in the P factor notion. But as you get down to the level of the factors, there's some differentiation at a biological level. And then one might imagine that with the accumulation of experience-dependent epigenetic effects, developmental, programs of gene expression, et cetera, and then of course environmental exposures and experiences, we might, have a kind of canalization into more specific clinical syndromes that we recognize in clinical practice and more broadly. Now, let's talk a little bit about the methodological caveats. And here's the challenge we face, and welcome comments from people. One is, and this is a paper I'll point folks to if you're interested in these issues, led by Na Kai, where we looked at, some of these challenges. But for example, these phenotypes, as I mentioned, are a moving target. And to the extent that they're not that reliable, what are we seeing? I pulled out here the um, inter-rater reliability from the DSM-5 field trial. So what they did is they had people go out, use the new DSM-5 criteria, interview patients, and two psychiatrists 2 weeks apart. And it's not exactly a great picture. So for example, these are kappa values. You'd like to see them be pretty high, close to 1 would be amazing if they were. But look at something like, so for post-traumatic stress disorder, it's actually pretty high. Bipolar disorder, we're getting down to 0.56. When you get down to generalized anxiety disorder, this is really very weak. There are also other issues. So what about the variation? We're putting together a lot of data, variation in case ascertainment. Some of these people came from or participated in healthy volunteer biobanks. Some were maybe ascertained from clinics. There's also the fact that they might vary in comorbidity. Sometimes specific comorbidities were screened out. And uh, to the extent that there is really shared genetic signal that might be causing comorbidity, pulling those specifically out of the cases might bias the results. Then, of course, there's variation in the control ascertainment. Some studies use supernormal controls. So they, screen out everything in the controls, maybe all of the disorders. That, of course, would deplete all of these other conditions and should inflate the apparent genetic sharing. Uh, then there's been differences in phenotyping depth. So some people or some studies have done very lengthy diagnostic interviews, documenting every possible variation in the phenotype, and others like, have used online surveys. Have you ever been diagnosed with depression by a psychiatrist or something like that? And actually, this has been looked at to some extent quantitatively. This is again from a recent paper led by Nakai and colleagues that looks at the genetic correlations uh, based on the kind of ascertainment. And you can see some of them are fairly striking, right? So self-report, look at ADHD. Uh, self-report compared to a clinically ascertained sample. Occasionally, uh, the clinically ascertained has a higher genetic correlation, so schizophrenia and bipolar disorder. This was ADHD and bipolar disorder. But you can see there's a lot of variation there. Another caveat is that our analyses were restricted because of just the diversity and size of samples to European ancestry samples. We did look at some cross-disorder genetic correlations in the samples we had, which were mostly reassuring, but it's not really a definitive test of certainly the structure of these factors, et cetera. Could misclassification actually account for all of this? The answer is probably no, it doesn't account for all of it. These are some calculations that were done in that Brainstorm Consortium paper that essentially calculated what would be the proportion of misclassification that would be needed to produce the observed genetic correlations between, pairs of disorders. So you would need to have misclassified a lot of people, not impossible, but in some cases for bipolar disorder, schizophrenia, uh, anxiety disorders, and MDD. We also looked at this issue of the phenotyping depth. How are we doing on time? We're doing okay. So in a sensitivity analysis, removing these questionnaire-based or very lightly phenotyped samples and then refitting the model. And what you see is in the bold italics, the estimates after removing, and these were the estimates before removing them or including them. And I think you'll agree it's not very different. Okay, why does this matter? Who cares? Well, one thing, and this was sort of motivating us, I think, uh, is we want to understand the genetic architecture of psychopathology. What do we learn about how what we think are these very important clinical syndromes are related at a biological or at least fundamentally genetic level? Maybe that could inform new additions or iterations of the DSM. And there is, in fact, a new iteration underway. And there have been some papers written about, how do we try to incorporate more perhaps biomarker data or etiologic data. It might explain something about the range of and massive amount of comorbidity that we see. Maybe we would expect some of this based on the fact that there don't seem to be very strong genetic boundaries among many of these conditions. And maybe we should be thinking of it as sort of underlying dimensions, actually, that transcend these conditions. And there's a lot of interest in that as well. Now, nosology or classification is about more than etiology. Even if we show that there are these shared, risk factors, that doesn't mean we throw out our diagnostic system. Obviously, here's a ridiculous example, but smoking is a strongly shared risk factor between COPD and lung cancer. That doesn't mean these are the same disorder. Childhood adversity is a powerful cross-disorder risk factor, explains a lot of the variance, also, across disorders. But again, that's not a basis for saying these are all just one thing. And of course, in the area of autoimmune disorders like inflammatory bowel disease, we see a lot of genetic sharing as well. So diagnostic categories have a lot of other functions that, you need to take into account. But knowing something about these relationships ought to inform our thinking about it as well. And that the other possibility is that it might have implications for our developing treatments. So FDA and the European Medicines Agency rely on the DSM for determining drug indications and approvals. It is, that's a huge impact of the DSM. They don't typically approve treatments that are for symptoms or syndromes that are, not previously accepted or defined. But maybe we should be looking at a more biological level as we think of identifying novel targets for treatment development or repurposing of treatments that, might in fact have effects here. And I'll just give you one example, again, growing out of this work. So in this study, we took data from the 5 factors. This was slightly before that latest thing, but it's basically very similar factor structure. And then looked at the genes that these variants map to using transcriptomic profiles and imputing transcription in the brain, and identified 466 genes across these factors. And then you can match them to mechanism of action based drug gene pair databases to say, are there compounds that are known that target the genes that are implicated here? And so, for example, in this case, we identified 39 known compounds, some of which are approved drugs, that would be predicted to be potentially repurposable for the treatment of psychiatric disorders, most of them related to schizophrenia and bipolar disorder, including L-type calcium channel antagonists or calcium channel blockers, which have previously been implicated in the genetics of those disorders. And they've actually been studied a little bit with, I would say, very mixed success. But you see the concept of how we might go from learning something more fundamentally bottom up to actually informing the possibility of treatments that are more aligned with perhaps the underlying biology. This is a quote from Sir Francis Walsh, a neurologist, which I think sums up the takeaway here. And I think that is actually potentially my last slide, which gives us a little bit of time for questions. And so thank you all for your attention, and look forward to hearing your questions.
Jordan, Dr. Smoller, thank you very, very much for this really fascinating overview. And 20 years of genome-wide
Mm.
association studies, GWAS work, what an enormous, enormous evidence that has provided. It was a wonderful overview. Thank you very much. I also thank you very much for bringing up Emil Kraepelin. Kraepelin would be, by some people, be
Mhm.
considered to be the precursor person for the DSM. He did the first, made the first classification, a psychiatric classification, was of course an academic arch enemy of Sigmund Freud in the same time. And I bring this up now, he died 100 years ago in 1926. You mentioned it. It
Oh,
was
right.
exactly. So we should perhaps not celebrate it, but at least take
Honor.
that as
Yeah.
a moment of interest
Mhm.
in his work. And I know this because he is the boss of Alois Alzheimer, and therefore I have studied
That's
also
right.
Kraepelin a little bit. It's wonderful. Thank you again. Now, normally I would open it immediately for discussion, but in this particular case, I would like to give the floor to a person who you mentioned, and I do this for two reasons. Number one, Dr. Debla Brecker is so important for us because she's the deputy chair of the Department of Epidemiology and very, very influential. And also has been very much in your path, Jordan. So, Deborah, I
Mhm.
don't want to put you on the spot, but
I'll say
if
I was
you
in
have a
her path
question, a
because
comment, or whatever you want to say, please do now. Yeah.
I
But
like in
you're
your path as long as you don't interpret it as in your way, which I'm like, I don't want to take any credit, but not too
okay.
obstructionistic. But Jordan, that first of all, that was absolutely lovely and incredibly interesting. And what I love about it is that it's both incredibly thoughtful about what the issues are. I mean, you like given that this is epi and you're talking about feeling like you've come home, it's very clear that you haven't left. So that's the really great part. Like this is really thoughtful about all the things that we think about in epi that you don't see enough of in some of the psychiatric genetics world, as I know you're very familiar with. Um, I wanted to ask one specific question because I was really impressed by your looking at the threats to validity and what the issues are and the idea of sort of seeing if the factor structure of the genetic influence could tell you something. But it seemed like the next step, and maybe you've already done this and it's a dead end, it wouldn't shock me if it were, but one of the reasons for the overlap is error. And, the kappas are terrible. But one of the reasons the kappas are terrible is that the symptoms overlap a lot across these disorders. And to me, the question is, how does the factor structure of the phenomena overlap with the factor structure of the genetics? Because then you're not stuck with these disorders that have arbitrary boundaries. I mean, the boundaries are not absolute, they're not random, but they are
Right.
arbitrary and they reflect political processes and financial incentivizing and all kinds of other stuff. So if you could look, I mean, symptoms are not perfect, but they're a little better, and they're also probably what our drugs are targeting, much as the
Mhm.
FDA claims that they're not. And so if what happens if you try to do that?
Yeah, that's such a great question, of course. Uh, uh, So the answer, well, partly I would say that, as you might imagine, the factor structure does depend on what you're inputting. And so if you know, throw in three other things and maybe leave one of these out, you get slightly different factor structure. There is now a lot of effort and now some fairly big studies um, on the using these kinds of methods to look at symptom relationships and particularly at the level of we often think of in psychiatry these foundational domains of internalizing and externalizing symptoms. So internalizing are those things like anxiety, depression, et cetera. Externalizing more in the realm of sort of substance use and behavioral disorders and so on. And so we do see some, as you might expect from what we saw in these disorders, uh, uh, separation. In this analysis, we did look also at how these things correlate with the disorders. There hasn't, to my knowledge, been um, a sort of comprehensive look at if you threw them all together, the disorder labels and the symptoms, what would you get? I think that I agree with you, though. It is more likely that transdiagnostic symptom dimensions are going to be where a lot of this is coming from, and that we might resolve some of the questions by looking at that. It does look even within, for example, work led by Travis Mallard and the Externalizing Consortium, uh, even within domains like that, there seem to be sub-factors that distinguish different kinds of externalizing behavior. So it becomes complicated, of course. I
Oh,
mean,
yeah, and I think that you're torn between whether doing things that are hypothesis driven or neutral, because either
right.
way, you have problems. But I mean, you could take out like the environmental variance drives some of that, and can
Yeah,
you take some
yeah.
of that out, the childhood adversity kind of thing? Although some
Mhm.
of that is parental psychopathology, that
And that's
that's
been done actually. So an analysis of the sort of factor structure effectively, or a network genetic correlation, removing the childhood adversity,
hmm
um, does reorganize the factor structure somewhat.
yeah,
Yeah.
yeah.
Can
But
I
the,
can I
but
do a follow-up question? I had a similar type of question where you were talking about the phenotypic misclassifications. As an amateur, I thought, well, two types, and if it's non-differential, then you would expect even a higher type of link between. So what you effectively say is that it's differential. That is to say, if you say the point 0.7 of major depression and schizophrenia, that people who are classified as depression, major depression, are actually schizophrenic, and the other way around. So that is basically what is going on, isn't it,
Mhm.
these analyses when you have misclassification, isn't it?
Yes. There's another phenomenon which I think is happening in Alzheimer's disease, which you may know better than I, uh, of as samples get bigger, their heritabilities decrease. So this has been documented in Alzheimer GWAS, where, adding more and more cohorts, adding sort of probably noise and maybe light phenotyping. And, in Alzheimer, you have proxy phenotypes as well.
Yeah,
Um,
yeah.
and so one of the phenomena we saw was that as you went to successive cuts of the data and larger samples, the genetic correlation between depression and PTSD started to almost become one. And so there
Mhm.
was you have
Mhm.
that secular trend in non-specificity of diagnoses in the samples you're looking at as well.
Yeah, yeah. Thank you.
somebody should feed that back to the head of some of the big genetics consortium, because there is a very substantial focus on the N without regard to quality and
Oh,
sample
yeah, yeah, yeah,
issues.
yeah, yeah.
It's really an important point. It's-
Well, on the
Exactly.
positive side, in genomic studies, that's why I think GWAS have been so successful, is that there are less issues generally of confounding even of selection bias, and of course you the case ascertainment and the control ascertainment is important, but there is less of an issue there as if you study non-genomic factors, huh? May I ask whether there are other questions in the audience? Comments, perhaps? I see none. Deborah, from your side, I interrupted you, I realize. So from
Well,
your
and I mean, I
side, a
could talk
further
about this
one?
forever. I don't want to bore folks, but I think seeing something about the structure and sort of large, like individual symptoms are hopeless, but I do think the sort of internal externalizing and psychosis kind of level things, and you probably have a data-driven thing that looks like that, but is more better articulated, and seeing whether you can get some teasing apart of genes, and it makes sense. The part that I hadn't heard at all, because I haven't kept up with this as well, is the incredibly interesting thing about how the things clustering on developmental genes,
Mm.
which
Hmm.
makes a lot of intuitive sense.
Yeah. Yeah.
So,
Can
but
I
it's
can I
very
ask
exciting.
Solorosito, Maria, would you ask your question? You put it in the chat, but would you ask it directly, or shall I read it for you?
Yeah, sorry. How can a psychiatrist diagnose a new patient who is already medicated by other doctors? I mean, the new doctor knows the patient that was already medicated, so they don't know like their original personality. If there is a way to explain that, and will genetic testing help with that?
Good question. You're right that it can be challenging. The way we approach it in that case usually is by history. So you try to get a complete history of when did the symptoms develop. Let's say, what were they like? You might get records from elsewhere. You might get collateral information from a family. So you try to put together the best diagnostic approach you can, despite, that people are somewhat treated. In some ways, that's not all that different from conceptually from other conditions where people are treated, their symptoms improve, but you could still make the diagnosis retrospectively. In terms of the question of genetic testing, I'd say the answer is no at this point. There really is no genetic testing that is clinically useful for diagnosing common psychiatric disorders. And there's some argument about whether that will ever really be the case. As I showed you before, there were some, for example, in the case of schizophrenia, there are some rare variations that have a very strong effect or high penetrance, relatively speaking. And those things are testable, or at least some of them, like that 22q11 deletion, that can be tested for. It affects maybe anywhere from 2 to 6% of people with schizophrenia. And it's usually associated with other syndromes or other features of a syndrome. So it's not the kind of thing that's widely used, but for things like anxiety, depression, bipolar disorder, really, no, I would say is the answer.
Thank you.
Thank you very much. I would like to make two final comments. One is to invite you to, in 14 days, not next week, but in 14 days, to a seminar again, and that will be a seminar of Elizabeth Arias. She is of the National Center, in fact, the head of statistics in the National Center for Health Statistics, and she has been permitted by our government to give a talk about the US Life Table program, the Life Table as the mother of all survival analytic techniques. So I invite you very much in 2 weeks, same time, same place. It will be on Zoom again. Second comment, Jordan, this was wonderful. Thank you so much for this seminar and for answering our questions and comments. Very, very much appreciated. And I hope you collaborate further and go on with this great work. Many, many thanks.
Thank you so much for having me and for everybody coming.
See you all. Thank you. Bye-bye.